Welcome, everybody. In fact, a very warm welcome from inside to those from inside and outside D-BSSE for today's eSymposium around the question of engineering and biology, where are we? Which I have the pleasure of co-hosting together with Mustafa Khammash and with the support from Elvin and Caroline. As coming together for our regular Tuesday D-BSSE seminars is a bit inappropriate in these times of Corona, Randy Platt introduced this format of the eSymposium, which allows us to discuss a specific topic a bit more in depth with people from inside D-BSSE and a main speaker that we invite to join us from outside. And I think it's fair to say that we made a real catch for our symposium today, when we could convince Wendell from USASF to join the program today. This is the second symposium that we hold after the first one earlier in the semester about the spreading of the coronavirus. And I'm going to say a few more words on how we plan this afternoon to develop. But before I do that, let me quickly say that if you like this format of the eSymposium, we invite you to stay connected by using either of the ways shown on the slide: email, newsletter, Twitter, or LinkedIn. And one more point, although I think it might be clear to everybody by now, we do record this symposium. And the recordings of this symposium, the one we had already, and the future eSymposia are generally available through our homepage. Okay, enough of the organizational chit-chat. This is what we're going to see today, although with a bit of a delay in the timing, which I hope you will forgive us. We're going to first have a set of talks from the lab of Mustafa, for a total of approximately 25 minutes, so no questions in between, followed by a first moderated discussion. And for this discussion, all of you should have available now a Q&A icon, question and answer icon in your Zoom window. And we invite you to place your question to either of the two speakers into the Q&A section already during the talk or at the end. You all can see the questions, and if you think that one question is particularly interesting or pertinent, please use also the chance to vote it up. And in this way, we hope we can make the most of the limited questioning time at the end by answering those questions that are of most interest to the largest number of people. If your particular question remains unanswered in this process, we're very sorry, but of course, don't despair, send a mail to the speaker directly. After this first Q&A, there will be a second set of talks, this time from the Bioprocess laboratory, again for 25 minutes, again followed by 5 minutes of Q&A. And then we will give the floor to 45 minutes of uninterrupted scientific excitement and leave the screen to Wendell, who's spending his breakfast time with us from his home in California. So thanks very much for taking the time, Wendell. Then without too much further ado, let me stop sharing so that Mustafa can take over. And let me take the opportunity to introduce Mustafa properly. I trust most of you know him, as he's of course from our own department. And obviously Mustafa is a perfect choice for this particular topic of an eSymposium because he is a hardcore engineer by training, so he knows the topic firsthand. In fact, Mustafa trained as an electrical engineer, first at Texas A&M and then at Rice University, from where he got his PhD in 1990. After that, he moved to Iowa State, founded the Dynamics and Control program, and headed that until 2002, when he joined the Mechanical Engineering Department at UC Santa Barbara. And then in 2011 was the critical year in his career because we could convince him to join the D-BSSE, where he's now heading the Control Theory and Systems Biology Laboratory. So Mustafa, we're very much looking forward to your talk.
Thank you very much, Sven, and thanks to all of our listeners all over the world. Good morning or good afternoon, whatever the case may be. It's my pleasure to welcome you to this eSymposium that I had the pleasure to co-organize with Sven Panke. And I'm going to give the first presentation on the topic of adaptation. So let me start by sharing the screen. Hopefully you will see this. Okay, here we go. So my topic is self-adapting circuits theory and implementation. Adaptation is a central theme in biology, as you all know. Homeostasis is a concept, strategy that can be seen at every level of organization in biology. And in my talk, I'm going to give you a synthetic biology twist to this very important concept. But first things first, let's try to see what exactly do we mean by adaptation. So we say that a process adapts perfectly, and I'm going to focus on perfect adaptation, when the process's output initially responds to an external input and then returns back to a basal level. So here's an example. See if I can move the screen. Okay, looks like my presentation is already stuck. I'm going to start it again. That's not, okay, here we go. So you have a system here that is subjected to some perturbation, perhaps a stimulus from outside, and there's an output of interest that we're observing. And assuming that the disturbance comes in at a particular time over here, what you would notice in a perfectly adapting system that the output of interest actually, after a short transient, returns back to its pre-disturbance level, even though the disturbance persists. Okay, so this is in the presence of persistent disturbance. And similarly, when the disturbance takes on a new value. So this is what we would call perfect adaptation. Now, when this adaptation remains in spite of the fact that the system's parameters change, we call this robust perfect adaptation. So it's robust to the variations in the system parameters. This is not a new topic. It's been studied in biology a lot. So one of the classic examples comes from the work of Panke and Leibler, famous paper in Nature in '97, which builds on work of Howard Berg looking at bacterial chemotaxis. And one of the remarkable thing about this particular system is that regardless of the amount of chemoattractants that these cells see, as you can see over here, after a short transience, the system activity goes back to its pre-chemoattractant levels. Okay, and this can be seen even though the levels of chemoattractants are increasingly going up. And notice that the adaptation is there, although the adaptation time could be different depending on the amount of chemoattractant. And in this particular case, the system activity is the tumbling frequency for these E. coli cells. And E. coli uses a strategy to chemotax because they're too small to sense nutrient gradients. So they have to convert temporal gradients into information about which direction to move. One of the interesting things in this paper, actually in the abstract of the paper, is the following statement: is that adaptation property is a consequence of the network's connectivity and does not require fine-tuning of parameters. And so Panke and Leibler presented a model that explains this and showed that this is indeed a robust property and not finely tuned. Another example comes a few years later from the work of Alex van Oudenaarde, who was looking at nuclear hog concentration in the nucleus of yeast after adding salt to the medium. And as you can see here, regardless of the concentration of salt, the nuclear concentration of hog goes back to zero after some transients. An example at a different physiological scale is one that I have studied with a former student of mine, Hannah El-Samad, who's actually in the department of our keynote speaker, UCSF, and a physiologist by the name of Jesse Goff. We looked at mammalian adaptation, calcium adaptation. And one of the things that we have found is that in spite of very large disturbances in the amount of calcium in terms of being drawn from the blood, the blood plasma concentration of calcium goes back to physiological levels, about 9 milligrams per deciliter, in spite of the drastic increase in calcium supply or demand. Okay, so this is another example of perfect adaptation at a physiological level. In engineering, I actually, we do perfect adaptation all the time. We call it robust disturbance rejection. And I built this little robot, some of you have seen it, to help teach this concept to undergraduate engineering students already at UC Santa Barbara. So this is not exactly new. So basically we have a robot that can send ultrasonic signals, and then the signals get reflected, and based on the time difference between that signal takes to travel, the robot can figure out the distance with any object in front of it. And the goal is for this particular system to maintain a distance of 30 centimeters robustly, okay? So this is what it looks like. And so first we're going to start with a robot with 4 equal sized wheels, and it's going to try to maintain a distance of 30 centimeters to this booklet in front, and I'll show you what it looks like. And when you turn on the system, you can see that it actually adapts to 30 centimeters, right? So the next thing what I do is I change the wheels of this robot, so replacing the small wheels with larger wheels. And the whole point is to demonstrate that this adaptation property is actually robust. So we'll see this now. So one of the things you will see from this example is that even though the adaptation is the same for the different wheels, the timing for adaptation is different. For the larger wheels, it's much faster. So the system is much more aggressive. The control system is much more aggressive than with a smaller wheel. Nevertheless, at a steady state, the property is maintained for both systems, right? So this is that's the example from engineering. If you want to look at a very simple model for this robot, so you have F equals MA, and the controller that I programmed into this robot is very simple. This is the torque, and it's given by a function of the error, the difference between the actual distance and the 30 centimeters, the desired distance. And the interesting thing I want to point your attention to is this term, this integral feedback term. And so the robust adaptation in this engineering example, and also in the 3 other biological examples that I discussed, happens because these systems employ this strategy of integral feedback. So in the feedback loop, you have a term that is dependent on the integral of the error, x of s minus 30. And so because of this integral feedback, we have perfect adaptation, force, torque, friction. If you change the mass, if you change the motors, change the design, it doesn't really matter. The adaptation is still maintained. So this is the robust perfect adaptation in engineering. And one of the important theorems in control theory is the internal model principle, which basically says that this particular case, that integral feedback is not only sufficient for perfect adaptation, but it's also necessary. So it's actually required. And what we would like to do next is to see if we can actually build this using synthetic circuits. We could take this concept and build it using molecules. Life is very different in biology than it is in the world of robots. So here we have biomolecules. We may or may not know them. Usually we don't know many of them. The interactions are unknown, so there's a lot of uncertainty. We know that the dynamics are very noisy, they're very nonlinear, and there's often external disturbances that are coming in. So in spite of this hostile environment, we would like to build a control system, negative feedback controller, that achieves robust perfect adaptation using synthetic components. So in this particular case, the controller itself must also be made out of molecules. And we would like to augment this network of molecules with the original system so that the closed loop, the system altogether, achieves closed loop stability, tracks a variable of interest x sub n, tracks a desired set point r, so it does reference tracking, and it achieves robust perfect adaptation, meaning the output is maintained in the steady state at level r, regardless of the changes in the parameters and the uncertainty and the noisy dynamics and all of that. So that's the goal. Now, there are various fundamental questions related to this goal. So how can we determine whether a particular candidate controller, as somebody may propose, regardless of its size, achieves robust perfect adaptation? And which architectural features of this controller are necessary and sufficient for biomolecular controllers to achieve robust perfect adaptation? Another question is, can we characterize all robustly stabilizing controllers? Is there a way to characterize the topologies that give rise to these robustly perfectly adapting controllers? And finally, which controller topology achieves this with the minimal complexity? And here complexity, I mean the fewer number of molecules and reactions. So I'm going to first give you one controller topology that does the job, and then I will address some of these other questions. I will be able to address all of these questions. So in order to do this adaptation, we have to be able to measure and actuate. And actuation is done with a molecule, Z1, that's produced at a constitutive rate mu. Measurement is done through the synthesis of another controller molecule, Z2, whose rate of synthesis depends on the concentration of the molecule that we would like to control, X sub n. So here we have a sensor, here we have an actuator. And then to close the feedback loop with the control system, we have another reaction, which we call an annihilation reaction or a sequestration reaction. And essentially what that reaction does, Z1 and Z2 combine together, and they either mutually annihilate or they just basically mutually inhibit each other. They just sequester each other, so that they become dysfunctional. And so this is the key reaction that is required to close the feedback loop. So if you close the feedback loop in this way, we can show that actually this simple system does in fact implement integral feedback in the sense that the average value of the difference between Z1 and Z2 is given by the integral of this constant. This is our reference now, mu over theta, minus the average value of the output of interest. So why am I taking the expectation operator here? Remember, we're dealing with a system that is noisy. So we have a stochastic dynamics. And for that, we need to look at the population average. If you don't like the population average, you can look at a single cell, and the same thing holds true for the time average in a single cell because the system is ergodic. So this type of feedback we call antithetic integral feedback. You have two antagonists, Z1 and Z2, that have to work together to implement this negative feedback. And we can show that theoretically that this type of antithetic motif does exactly what we want it to do. It does the robust perfect adaptation in spite of the noise and in spite of the uncertainty. Let me now tell you a little bit about all other controllers that do this job, because now we can explore this further and we can ask the question, what other motifs, if any, can do the same job? So if we look at our system here with the controller, we have our sensing reaction, we have our actuation reaction, we have our set point reaction. And I'm going to look at the controller species. Assume a controller has m different species. M could be any number. It's not restricted to any particular value. And the stoichiometry matrix for the controller is given by S. And these reactions have some parameters. So controller reactions have k different parameters, one for each of the reactions. And we're going to assume that the closed loop system is stable in a stochastic sense. If the system is not stable, then there's no point to talk about adaptation. In fact, that's true for all feedback control systems. You know, this is why control engineers obsess about stability. First, you have to stabilize, and then you ask what kind of performance requirements you can achieve. So in this case, we're going to assume that this closed loop system is stable in the stochastic sense. And the system achieves robust perfect adaptation if the output of interest equals to mu over theta, equals to our reference, in spite of the uncertainty in the controller parameters and in spite of the uncertainty also in the controlled network. And so the condition for achieving this, actually a necessary and sufficient condition, is the existence of a vector Q in the left null space of the stoichiometry matrix. And Q has to have 1 at the first entry and -1 in its second entry. So if you think about this, this is just a purely algebraic condition. It's a system of equations, AX equals B. It's very easily solvable. So if you have a controller and you want to know whether your controller achieves robust perfect adaptation, you form its stoichiometry matrix and then solve this systems of equations. If you can solve it, then you have a certificate that your controller achieves this robust perfect adaptation. Otherwise, you know it does not. But is there any different way to look at this particular condition? It turns out that you can unravel this condition further and get an internal structure of all robust perfectly adapting controllers. And this is this result. So every controller that achieves robust perfect adaptation must embed in it an antithetic motif, like the one I showed you before. So what does that mean? It basically means any controller that achieves robust perfect adaptation, you look at the controller species, they can be partitioned into 3 classes, C-, C+, C0. And there has to be one reaction that takes a molecule from C-, combine it with a molecule of C+ with a product in C0. So that's what we mean by embedding this controller. And what you can see immediately from this result, that the actual controller I showed you in the beginning is actually the minimal controller that will do it, because it only has 2 species. And you need at least 2 species to achieve this robust perfect adaptation. So in fact, we know what they all look like, and we know what's the minimal one. Now, it's important to emphasize is that not every controller that achieves robust perfect adaptation in the deterministic setting, so in the noise-free case, if you don't have noise, they don't all have to look this way. So this is not a necessary structure for perfect adaptation, for robust perfect adaptation in the noise-free case. But it is sufficient. So in a noise-free case, this controller will still work, okay? All right, so can we build it? That's the last couple of slides. I would like to show you how one can implement such a controller. So I'm going to show you our implementation in E. coli. So we have here the reaction mu. So here is produced by a sigma factor, SIGW, which is expressed constitutively. We can change the expression rate by adding HSL to this circuit. So this allows us to adjust the set point. And now SIGW drives this gene that produces RSC with the SFGFP. RSC in turn activates the anti-sigma factor, the gene for anti-sigma factor RSIW, which then combines with the SIGW, and they bind very tightly. And in that configuration, SIGW cannot reactivate RSC. So this is our closed loop system. In order to verify that this system actually does what we want it to do, we have also engineered a circuit that expresses MFLON that degrades RSC and SFGFP, and we can control the amount of MFLON by adding ATC. So altogether, we have 7 genes, 6 promoters, and 2 plasmids for this circuit in E. coli. And the control, or the open loop without the feedback, essentially we don't have the anti-sigma factor RSIW here. So we just have, you could turn on RSC and you can degrade it, but you cannot close the feedback loop. So here's what the results look like. These are results for different set points. You could see here in the open loop, when you add ATC, the ATC, the MFLON will degrade RSC, and you see its levels will go down by almost 50% in every case. In contrast, in the closed loop system, we have perfect adaptation. Although degradation still happens because of the feedback loop that's perfectly adapting, we have more of the RSC being expressed to compensate completely for the rate of RSC that's being degraded. You can see this also in the time domain. So here in the open loop, once we add ATC, you could see, this is without ATC. With ATC, you could see that the concentrations go down over time. And in the closed loop, once you add ATC, you have a small transient and then steady state tracking error that is barely measurable in this case. Finally, we could apply this to a growth condition to control growth rate of E. coli. And the way we do this is by now putting MET-E, an enzyme that's required for the biosynthesis of methionine, an essential amino acid. So we delete methionine we delete the endogenous MET-E and remove methionine from the medium. And therefore, we can make the growth of these cells under the control of our circuit. And by changing the set point, you could see that the growth rate will be different. And for the same circuit, when you change the temperature from 37 degrees to 30 degrees, without the feedback controller, the growth rate drops dramatically, more than 60%, whereas with the feedback controller in place, the growth rate remains unchanged. So this is just a proof of concept that these type of circuits can actually be built, and one can, in fact, realize robust perfect adaptation using molecules. What remains to be done next is we would like to improve the performance of the circuit, so the transients. So you can have robust perfect adaptation, but maybe the system takes a long time to settle. You can make it faster by adjusting different parameters in the controller. So this is more like a transient response. That brings me to the end of my talk. As a conclusion, I have shown you a family of universal self-adapting circuits, each member of which achieves robust perfect adaptation. And this is done in the presence of noise. In fact, what I haven't shown you is that these type of controllers even exploit noise, so that in the noise-free case, you may have instability. The fact that noise is present is exploited by this controller to stabilize the system. And this is an example of robustness through architecture. So you don't have to worry about tuning the parameters. You get the adaptation just by getting the right architecture. And this is important for synthetic biology, obviously, because you don't want to be tweaking parameters to get the functionality that you want. And so this is also an example of the first integral feedback in a living cell implemented with molecules. The one thing I would like to emphasize is the role of theory in all of this. Before looking at the antithetic, we spent a good 2 years trying to build a different controller that really failed. And it was only after we looked at this antithetic theory that we started getting designs that work. And this allowed us to discover this antithetic motif, which is non-obvious, but it turned out to be the right design for this task. And in the process, it also helped us elucidate biological design principles. There are various applications for industrial biotechnology and cell therapy, but that's another talk for another day. I have some references here for those of you who will go back to the video, would like to dig more into this. There's a lot of work on this, mostly in the deterministic case, some of it actually by our also keynote speaker who looked at 3-node networks and explored the different topologies among 3-node networks to achieve robust adaptation. Here's my group, and here are the people that did the work. Ankit Gupta did this characterization of all robust stabilizing controllers, and together with Corentin Briand came up with the original motif and the proof. And the circuit was built by these people, particularly Stephanie Aoki and Gabriella Lelacci. And with that, I come to the end of my talk. And I thank you for your attention. My understanding is we're not going to take questions now, so I'm going to then stop sharing this and have introduced to you the next speaker, who is my student, Timothy Frei. So Timothy started his undergraduate studies in pharmaceutical sciences at ETH Zurich, but then decided to switch to the Basel campus, to D-BSSE. In 2016, he finished his master's and joined my group as a PhD student, so he's a 4th-year student. And he's focused his work on theoretical and experimental approaches to develop and implement novel genetic circuits in mammalian cells. He's going to tell you today in 10 minutes or so about his work on burden in mammalian cells, so how synthetic circuits must share resources with the host and the implications of this resource sharing. So Timothy, It's all yours.
Thank you, Mustafa. And hi, everyone. It's my pleasure to give you a brief glimpse of some work that we've recently published in Nature Communications. This is work that we've done in collaboration with Velia Siciliano's group in IIT in Naples and Guy Stan in Imperial College in London. So one big problem that we still face in synthetic biology and in particular in genetic circuit engineering is that our parts that we use to build these circuits usually behave quite differently in isolation compared with when we put them together into bigger systems. So in synthetic biology or in biology in general, I think we call this context dependence. And so a while back, we did a very simple experiment where we co-transfected two plasmids that constitutively express a fluorescent gene. So one plasmid expresses the yellow-green fluorescent protein M-citrin. The other one expresses the red fluorescent protein mRuby3. In this co-transfection, what we did was we kept the level of one of the plasmids constants while we increase the level of the other. So in the sort of first scenario I'm going to show you, we fix the level of mRuby3 and we increase the level of M-citrin. So if we do this transfection and then measure the gene expression with flow cytometry, what we see is that, as expected, our M-citrin levels increase as we increase the plasmid amount that we transfect. However, a bit surprisingly, the constantly held mRuby levels decrease as we do so. And so this, in this sort of setup, you can think of the constant levels of mRuby sort of as your context. And so in a sort of next step, if we change the context, we can also see that, so if the context increases, there's also a decrease in M-citrin. So for a constant amount of M-citrin levels, there's a decrease in M-citrin. This is also true for other levels, and it's a very consistent effect. And so the way we sort of rationalized this observation was by looking at resource burden. So this is something that is already has been well known in bacterial synthetic biology, but has not been well described in mammalian cells before. So what exactly do we mean when we talk about resource burden? So in general, it's competition for a limited pool of resources, which in that effect creates an indirect coupling. So as I'm illustrating here, so I should also mention here, I'm displaying this finite pool of resources as ribosomes, but I want to stress that we at this point do not know exactly what pool is limiting. So I'm just taking this as an example. So in this example, we have some level of mRNA, for example, for a green fluorescent protein, which is consuming resources in the process of translation. Now, if you introduce another gene, for example, as we saw before, the red fluorescent protein, this newly introduced mRNA will now draw away resources from the translation of the green fluorescent protein into its own translation, and in that effect, reduce the expression levels. And similarly, along those lines, if you keep increasing the levels of the red gene, more resources are drawn away from the production of the green fluorescent protein and towards the red fluorescent protein. So this is a mechanistic way of how we sort of rationalize the observation that we had. Now, in general, so this, as I said before, is already something that was well known in bacterial cells. So here are some references to studies that have been conducted over the years, and you can see there are quite a few. Now, in general, people usually only consider a single pool of limited resources, but we sort of considered that since we don't really know which pool or what is limiting in the process of gene expression, we sort of also hypothesized that the other major process in molecular biology, so transcription, might also contribute to this coupling effect that we see. So in the publication, we have some evidence for this, but unfortunately, due to time constrictions, I have to limit, or I can't really show any data on that. Nevertheless, so in synthetic biology now, to really sort of solve this issue, so this decoupling between the expression levels is something that we don't really like, because this causes our components to interact in ways that we don't necessarily intend them to. So for doing so, we came up, or we consider what is also something well known in synthetic biology, which is this incoherent feed-forward loop motif. And the way this works, so I'll take you through the experimental setup that we have. So we have a constitutively expressed fluorescent protein, which here is MIRFP, that consumes resources in the process of being expressed. And then we have a separate plasmid which encodes two constitutively expressed fluorescent proteins, mCitrin and MRUB3 as before. But now MRUB3 intronically encodes a microRNA, more specifically the microRNA FF4. Furthermore, both of these genes are tagged with targets for this microRNA. So in the process of being transcribed, this plasmid produces a certain level of mRNA and the microRNA. And this microRNA, because both these mRNAs are tagged, will inhibit the translation. Therefore, so you can sort of consider that while we have 4 mRNAs here, only 2 of them will be translated and produce protein. Now, when we increase the amount of MIRFP plasmid, more resources are drawn away from the production of these mRNAs, and similarly, resources will be drawn away from the production of this microRNA. So now, again, in this process, the repression of the microRNA is alleviated, while also the amount of mRNA that goes into the translation is reduced. And therefore, sort of on average, you can argue that the gene expression will be somewhat unchanged. There's other effects that still affect this, but I won't go into any details on that just now. So in the actual experiment that we did, we had a control where we had the 2 genes tagged with mismatched targets, and similarly as in the first experiment that I showed you, we increased the amount of MIRFP. So if we do so, you can see that the levels of MIRFP increase, which is what we would have expected. And in accordance with the effect that I described before, both the levels of mCitrin and MRUB3 decrease. Now, if we do this with targets that are matched to microRNA FF4, we can see that again, the levels of MIRFP increase when we do this titration, but both the levels of mCitrin and RUB now decrease less than before, therefore sort of giving us some evidence that the decoupling has been reduced. So this is a nice kind of application to sort of, if you have components in a circuit that you would like to insulate from each other, maybe an incoherent feed-forward loop can do that for you. So with that, I already want to mention, or I want to shamelessly plug our publication, because there's a lot more that we did that I didn't have time to talk about today. So we provide a lot more evidence for this effect in mammalian cells. Federica, which is co-first author on this publication, has done a very nice characterization of microRNAs. We have a mathematical framework that can model these effects. And Federica also managed to show that you can use endogenous microRNAs to get this mitigation effect I just described. I also want to point out another recent publication that does something very similar in a very similar setting. So if you're interested, please go check them out. And with that, I want to thank everybody involved. And yes, please get in touch if you have any further questions.
Thank you very much, Timothy, and thanks very much, Mustafa, for these very fascinating insights into how to build controllers and how to manage metabolic systems, mammalian systems. I'd like to point everybody towards the Q&A function for posing and maybe upvoting questions. In the meantime, let me put two, one for Mustafa. What molecular systems can achieve antithetic feedback? Here you use anti-sigma, stoichiometric complexes, but what are the requirements in terms of affinity, concentration, dynamics, and what other systems can fulfill this?
Okay, thank you very much, Sven. So the requirements for antithetic control are in some sense rather strict. So the reaction that is responsible for annihilation, so Z1 and Z2, must disappear stoichiometrically. So one molecule of each or two molecules of each must disappear at the same time. It cannot be a catalytic reaction, so it cannot be that Z1 degrades Z2, but still lives on to degrade other molecules. So that eliminates things like microRNAs and so on as molecules for achieving adaptation. Now, the affinity, usually these things should have high affinity, but they need not actually, doesn't need to be too large. It could even be reversible. So the binding and unbinding could be reversible, so long as when they're bound together, Z1 and Z2, there is degradation of the pair. Not one, but together they have to degrade. I should also say another requirement for adaptation, for perfect adaptation, is that Z1 and Z2 need to be stable molecules. And this is perhaps the most severe requirement. And so in E. coli, this precludes pretty much, using mRNAs for or sRNA for achieving this task. They don't need to be completely stable. They just need to be, the degradation needs to be slow enough relative to the dynamics of the process that's being controlled. So that, for example, in this case, we needed to use proteins, sigma factors, anti-sigma factors, because of their stability, but there's still dilution going on. These cells are growing. The dilution rate was not so large that it destroyed the perfect adaptation. Now, what happens if these Z1 and Z2, these molecules do degrade? Well, you have what we would call a leaky integrator. It's basically you're trying to fill a tank, and the integrator, if you think of a flow rate as the input and the level of the tank as the integral of the input, which is what you need, a perfect integrator. If you have a hole at the bottom of the tank, you will get a leaky integrator, which would give you a small steady state tracking error. So the more stable, the better, but you just need to, these molecules need to be stable relative to the process being controlled. Just one last comment to this question, so we're just starting to look at natural systems. So we found, for example, in the case of sigma 70, there's an anti-sigma 70 that appears to be implementing this type of feedback loop in bacteria. We also have some evidence that even in signaling, this motif might be there. For example, sonic hedgehog signaling and the patch receptor may be performing this function. And this is intriguing. It's still work that is being carried out, so we're still exploring that. But anytime you have two molecules that antagonize each other, that are in a feedback loop, there's a very good chance that what is going on is some type of a perfect adaptation loop.
Okay.
Thank you for the question.
Then one for Timothy, very nice talk and project. Adding the feed-forward motif is reducing the total amount of fluorescent protein, question mark. And did you think your finding can be translated into bacteria?
Yeah, that's absolutely right. I sort of skimmed over that. There's a price to pay, and the price is the expression level of your protein. So if you introduce this feed-forward loop, then you reduce your expression level by quite a bit. For introducing this into bacteria, I haven't specifically thought about that, but if you can find a similar mechanism, which I guess, for example, small RNAs in bacteria would be, and they both, so one requirement for this to work is that both of your, so the small RNA and your RNA need to be produced by the same resource. So they need to see the same pool, so that essentially the interaction with the small RNA can cancel out this coupling effect that occurs. But so if that is the case in bacteria as well, then I don't see any reason why one couldn't attempt that as well.
Okay, thank you very much. Question time is over, and the anonymous attendee who put another question in there, please feel free to send it directly to Timothy. And with that, we close our first round of talks, and I give the floor to Mustafa.
Okay, thank you very much. And it's my pleasure to introduce our next speaker, Professor Sven Panke. Sven is a full professor at the Department of Biosystem Science and Engineering at ETH Zurich, that's located in Basel. And he heads the Bioprocess laboratory there. Sven received his diploma in 1995 in biochemical engineering from the Technical University of Braunschweig in Germany. Then he received his PhD from ETH Zurich at the Institute of Biotechnology in 1999, after which he went to industry for a couple of years, where he was a biocatalysis research scientist at the DSM Pharma Products in the Netherlands from 1999 to 2001. At that point, he returned to ETH as an assistant professor, this time in the Bioprocesses laboratory in DMUFT, in the Mechanical Engineering Department, where he stayed until 2009 when he joined our department, a young department in Basel. So he's received several awards, including the DSM Research Award, the ETH Silver Medal for excellent teaching. And in his lab, in his bioprocesses lab, they revolves around the design of novel bioprocesses for the pharmaceutical and chemical industry. He's very active in the field of synthetic biology. He's a mentor and a judge for iGEM. And his ultimate goal in this area is to convert biotechnology from a discovery science into a true engineering discipline. It's my pleasure to have Sven be our next speaker. Sven, take it away.
Thank you very much, Mustafa, for this most kind introduction. I feel a bit embarrassed, but nevertheless, thank you very much. And as the title of the symposium is about the engineering of biology and the discussion of how far we have gotten in this endeavor, it's maybe a good starting point to take a moment and go back to the origins of this idea of engineering biology, which is also one of the central ideas behind the foundation of our department, D-BSSE. So 20 years ago, around 2000, a new term appeared on the scene. This term was synthetic biology. And while the choice of the name has produced, I guess, hundreds of raised eyebrows ever since, the idea started to attract followers immediately, I would say. The idea that you could turn our often very poor attempts to convince cells to do something that was not useful for them, but for us, into a projectable, rational endeavor along the lines that classical engineers use to build things with novel functions. And I would like to use this picture here on the right, and I seem to be not really Okay. I'd like to use this picture here on the right. That I stole from Drew Endy, and what was one of the iconical pictures that we used over the first couple of years in trying to communicate what we want to do. To point out why this idea has been attractive to many people in the early days of synthetic biology. And the central idea is that you might be able to organize biological engineering along something that is known as the hierarchy of abstractions, so that biotechnologists could organize their efforts along specific ideas, designing complex functions, designing parts, proteins, for example, or designing intermediate complexity devices that you could focus on, and then you would not need to understand all the details that happen in the other levels of the hierarchy. And to top it all, at the bottom of the hierarchy, you just sit down, print your DNA with all the information you need, and ship it out to a synthetic DNA provider, get it back, put your DNA into the system, and it would all work magically. Just to give an idea of how alien this idea was to what people like me thought at the time was, let me share the philosophy of my the father of the first teaching program in biotechnology in Germany, Fritz Wagner in Braunschweig, who insisted that his students would need to learn everything from genetic engineering to product purification from a fermentation process, because in biotechnology everything was so interdependent on everything else that in order to get anywhere you need to know it all. So now it was this idea of interdependency versus idea of modularization of a hierarchy that could allow you to guide your efforts. Whether you agree with the concept or not, I think it's fair to say that there have been some successes over the last 20 years, and I took the liberty of pulling out my personal selection of some that I think are particularly interesting. One example here on the left from the lab of Jason Chin, who was able to come up with a full-scale recoded genome in E. coli. One example here from the D-BSSE from the lab of Martin Fusenegger, who implemented together with the modeling capacity of the group of Jörg Stelling, closed loop glycemic control. Here an example from the lab of Chris Voigt, who was able to go for the entire nitrogen fixation pathway through a combinatorial assembly that allowed him to reconstitute this in, for example, E. coli. And another, what I would consider a very nice example for the success of synthetic biology, we have just heard in the talk from Mustafa in Universal Controller for Perfect Adaptation. So if there are these successes, then why might the engineering of biology remain difficult? I think it would be fair to say there are two main points that we need to consider. So there are simply things that make biology very different from your standard motherboard or your standard car that you want to engineer. And among these, I think these two are central: complexity and evolution. Complexity comes in many forms in biological systems, and I just mention here as an example that despite our best attempts and hundreds of research papers to the topic, I think the idea of how to get a specific amount of protein from a specific DNA sequence in a cell is not really solved yet. We're getting quite good at specific aspects of it, but we're nowhere near anything that a mechanical engineer would really get excited about. We do not fully understand the sequence-function relationship, the dynamics, the structural biology, nor the noise. But the even more intriguing obstacle in my mind is evolution. Maybe we come to grips with complexity at some point, and we think we heard quite some interesting ideas in the talk from Mustafa. Evolution is unlikely ever to go away. It's part of the cellular nature of biology. Cells need to replicate the genome and the surrounding cellular content, and replication is error-prone. And I cannot see a path to get really fundamentally rid of it. To make things a bit worse, evolution is actually one of the main aces that we have up our sleeves if it comes to manipulating cells into what we want them to do. Let me cite a rather old example before the 1980s, so before genetic engineering took off. Directed evolution was essentially all there was in our hands in order to convince systems to do something different. And along with this, we got, for example, the titers in penicillin fermentations up from 5 milligrams per liter to 50 grams per liter, so by 4 orders of magnitude. Since then, with all the molecular wizardry that we have at our fingertips, today it has been a modest factor of 2. Also in our group, we use directed evolution to our heart's content. And let me briefly show this example here from Stephen Schmidt and Martin Held, who took our nanoliter reactor technology that you see here, alginate beads with cells to produce libraries of antimicrobial peptides, one of which you see here. We varied the various modules that you can see in this around 30 to 40 amino acid peptide. These modules were exchanged for others, etc. And we used this technology of putting bacteria into alginated droplets in order to find those that actually kill sensor cells as opposed to others that do not. And in this way, developed by directed evolution peptides that had novel properties, for example, novel levels of minimal inhibitory concentration against standard multi-drug resistance pathogens from the clinic relative to the starting strains. So why do we have a problem? If evolution takes us so far, why do we have a problem? And this is a very busy slide, but I wanted to be a bit transparent about the next statement that I make. And what you see here fundamentally is just a calculation of how many cells and how many generations you get when you start from a single cell of E. coli to a 10 cubic meter reactor that is full of cells, something on the order of 100 grams of dry weight of E. coli per liter. Then you need on the order of 60, specifically 61 generations. And if you do all the calculations, how much DNA you produce in these 61 generations, then, and multiply this with the error rate of the typical DNA polymerase in E. coli, you find out that every single nucleotide in E. coli gets, statistically speaking, mutated 3 times 10 to the 8 times. So whatever your idea of a mutation in E. coli is, you can be sure that on the way to a 10 cubic meter full of bacteria, making insulin or whatever they make, you have explored all of these mutations. And it's only a question of time until the one that is going to make your system less effective and the bacterium that produced this mutation more fit compared to the others, until this particular strain takes over. Inevitably, there's no real way around that. Of course, this has not kept many biotech processes from appearing, because there's also the element of time. Just because a mutation appears does not mean that it's appearing at the very beginning and ruining your entire process. But if you think of long-term stability, this is the problem that we're up against, and there's just no way out of it. One tech that we explored in order to get out of this problem is cell-free biology. no more replicating systems, no more noise, because you aggregate material from very many cells into one system. And the example I have here is already a bit older. It's an 11 enzyme reactor that we assembled from purified enzymes in order to make this particular compound down here, dihydroxyacetone phosphate. I'm going to spare you the details of why we were interested in this. The point is we set up this 11 enzyme system to make sure that all the cofactors we need in the system can be regenerated, and that the energetics work so that you can actually, in theory at least, can convert 100% of glucose into 100% of your product that you are interested in. And in fact, indeed, at that time, we could do quite some fancy MS analytics that allowed us to do track such a system over time. And doing the ODE differential equations, all this was done by Christoph Holt, we could come up with a pretty good model for the system that allowed us to predict how the system would behave. So cell-free allows you to actually come to predictable systems. And it also allows you to work on problems that are a bit more off the beaten track, in the sense that you work with stuff that is a bit alien to normal biological systems. So here we look at a project that we do together with Tom Ward from the University of Basel, and that was started by Markus Jeschek and then taken over by Philipp Rottmann. We look into enzymes that work on totally novel cofactors, and for these novel enzyme systems, you need very often at the beginning quite weird starting conditions that are not physiological, 500 millimolar magnesium chloride, pH 4, etc. You need to worry about things such as how to get specific enzyme cofactors across the membrane, etc. All of this is quite difficult. And if you go to cell-free systems, you might be able to overcome many of these initial problems until you reach a system that is more physiological. In fact, Philipp was able to build up a whole directed evolution microfluidic platform around this problem, running multiple steps, including droplet sorting systems. But in the end, I guess the big jackpot of it all is going to be a cell that looks substantially different, that will allow us to use evolution for the weird questions that we are going to ask. In my mind, these are questions such as, can we use alternative compounds in the system others than DNA, RNA? Can we use proteins with alternative amino acids? Can we get to novel templatable biopolymers? Some of these questions are already answered to some extent. The increase of the number of amino acids in the system, for example. But I think these type of questions we will most probably not get, we will not arrive at solutions to these questions without evolution. So how can we marry the two? And that's also my transition to Daniel, the next talk that we're going to have. Of course, there is an optimal strategy that would allow us to combine engineering and evolution. And the way we try to do this is learn from engineering, take the models that allow us to do evolution, but in a constrained search space. Use the models to say what is reasonable to expect, and in this way constrain the huge search space that we otherwise have for evolution. And here's an example from Daniel's earlier work that he's not going to talk about. I think, in the interest of time, I skip over it to give him more time to talk about his idea of using engineering to constrain search space. And before I leave the floor entirely to Daniel, let me briefly point out a couple of people without whose collaboration this would have been completely impossible. Most of them are already mentioned. I should mention Tanya, Markus, and Martin Held, who have been instrumental over the last years to do all these things, and I'm very grateful. And I also like to point out my gratitude to these 6 people here at the bottom that you, of course, all recognize: Kobi, Carsten, Jörg, Nico, Konrad, and Petra, who have helped us at some point getting further in these various endeavors. So with that, let me close and give the floor to Daniel.
Okay, can you hear me?
Yeah.
Great. Let me share my screen. So today I want to talk a little bit about my project I was working on during my PhD, and it's about synthetic operants. So as you heard maybe a little bit from Sven's presentation, in synthetic biology or in bioprocesses, we want to convert simple, cheap, renewable substrates into really interesting products for chemical or pharmaceutical applications using microorganisms. To do that, we often introduce synthetic pathways into a system to convert actually the substrate of that we choose into the product of interest in the end. But we need to do this all in a living chassis, actually, and this comes with a lot of problems. Like the microorganism needs to take up the substrate. There might be several interactions with the host metabolism that might be problematic for the growth of the organism. There can be accumulation of toxic intermediates or formation of side products. And one of the tasks in synthetic biology that we have is to actually balance the expression levels of all these involved proteins to streamline the flux towards the product of interest. What we often do when we build these synthetic systems in E. coli, for instance, we construct synthetic operants that consist out of one promoter under which several genes are controlled. One of the reasons to do this is it's definitely a simpler system than having multiple promoters with several regulators and several inducers. And often we also have a limited number of really well-characterized, well-working promoters in synthetic biology. The other thing, also Sven mentioned it, is genetic stability. That can be a problem once you scale everything up. A smaller, more compact system that doesn't have too many repetitive sequences like several promoters is also more genetically stable. But this is not something we came up as synthetic biologists. Operants are really a very common structure in bacteria. For instance, in E. coli, more than half or around half of all genes are forming operants with 3 or more genes. And it's typically these genes are used as a coordinated response to new nutrients. Maybe the promoter can react to a new nutrient and then the cell can metabolize this nutrient or react in another way to the environment. What you see here depicted in small semicircles are, for instance, ribosomal binding sites, how we depict them usually in synthetic biology. And Sven mentioned it before that I was working on RBS libraries to kind of find out better how to look into different expression levels. But once you build actually a whole operon, it's getting more complicated because the predictions are not as accurate anymore. And you're really kind of screening randomly almost in the blind. I'm depicting this here. What we often do to try to optimize these systems is really a lot of random sampling, which is not very efficient. What we would like to do in future is really go towards forward engineering, better understand our systems, and thus more efficiently sample the underlying performance landscape of the system of interest, and thus really speed up the optimization of these synthetic systems. The operant systems that I was working on, I'm having here a little diagram about this. The system that sorry, I'm having here my face in the way. So the system I based my synthetic operants on is based on the T7 promoter, which is a very commonly used promoter in synthetic biology, as it's a rather orthogonal promoter from a phage, and thus you can kind of separate your system a bit better from the host organism. I'm using a standard backbone and also refactored genes with synthetic RBSs to really make this system as unnatural synthetic as possible. But when you build synthetic operants, there's one question that might start at the that come at the beginning, because operants are kind of a polar system, and so you need to decide at which position you place the genes. And the gene order might actually have a big influence on the expression levels. And that's the first thing that we want to really investigate deeper. And I'm jumping really quickly already to some results there, because there was also a lot of how to assemble this in the whole project, but I will skip this now. And one of the data sets we generated was a data set of tricystronic operants expressing several fluorescent proteins, where we varied the gene order. You see this depicted in these letters, where each letter corresponds to one of the genes. And these gene orders were varied, and we looked at the expression levels of each of the fluorescent proteins. And if I just zoom in into 4 examples, we found that there are several expression level patterns that you actually find. So like an increase in expression over the position in the operon, or decrease, or a dip, or spikes within these expression levels. This was not done the first time in literature. What was done before were smaller tricystronic operant data sets, where sometimes you found this decrease in expression level, but I think we were one of the first that generated a larger data set that really revealed that there is potential for various expression level patterns there. Additionally, we then increased also the size of the operants, as there's also only limited data available about synthetic operants in literature. And we could really see again here that it's not straightforward. There's no straightforward pattern observable, like only increase of expression or decrease of an expression over the size of the operant, but we see various patterns like zigzag patterns, increase, decreases. Everything seems to be rather complex and highly context dependent, and it's hard to describe it with a very simple rule like we would like to do. We kind of, you can kind of expect this if you look a little bit deeper into how operant expression levels from operants is regulated in nature, because there are various elements encoded on the DNA, like RNA sites or structures. Once you have the RNA that can protect from degradation or increase degradation, and this eventually leads then to a distribution of various subsections of your transcript. This already leads to differential gene expression, but then on top of that, you have also translational coupling, which is a process how the ribosome, the translation from the ribosome of an upstream gene can influence the expression level of downstream genes, depending on structures on the RNA that might be inhibiting or increasing this effect. And all this, in the end, leads to various expression levels from one operon. And with rearranging the operants, what we did with different gene orders, we rearranged this kind of regulation, and thus generated really this variation of expression level patterns. However, we also wanted to try to explain how we find or build a model around these expression level patterns. With the data set that we could generate here, we eventually then came up with a machine learning approach where we used random forests to look into the various sequence features that are on the operon, and then train this data, this random forest with the data set that we have and all the various context-dependent sequence features. With this, we could already start applying this prediction to newly built operands and look into how well this prediction holds true. Let me zoom in quickly here. What I show you here is with the circles, the measurement data that we actually have from these newly built operands, and in the background, you see this horizontal bar of the prediction with a confidence interval. And for most cases, actually, we have a very nice prediction that's matching nicely the expression levels that we actually measured. I would say here I highlighted 24 out of 27 of these operands that have a good prediction. The yellow ones are having one of the predictions a little bit more off, which is still acceptable, but all these other green panels here show that our prediction is very close to the measured data, and really you see all kind of expression level patterns from the increase from the spikes and zigzag patterns, all is predicted well. Now, having this kind of predictive model, we can generate in silico all kind of permutations of operands. And this is what you see here. This is all 120 permutations of 5 genes and how the expression level patterns look over the length of the operand. And now as an experimenter, you can just choose which of the points in the performance landscape you would like to test to really see what is the performance, and choose only these operands to build and to efficiently test then how and efficiently optimize your system without having to really randomly sample all kind of permutations where you don't know the expression level beforehand. So I'm coming with this to my summary. What I skipped a little bit was that really thanks to automation, we could assemble a large variation of gene orders. We had up to 7 fluorescent proteins with which we could generate almost 3,000 data points. And this revealed that there's really a large diversity of expression level patterns. And thanks to our random forest model approach, we could successfully predict new operand designs and hopefully in future then help with this synthetic biologists to design their new systems in the first in their optimization approach. So I want to thank especially my mentors and supervisors, Sven, Marcus, Tanya. Also big thanks to the automation facility at the BSSE that helped me a lot building these operands. So thanks to Nico and Susanna for some helpful machine learning discussions. And I also want to mention that while we have now this machine learning approach, we actually want to in the end also understand what's going on the molecular level. And that's why we're collaborating with the group of Jörg Stelling, with the help of Claude and Jörg's group, to really find some mechanistic model that explains the expression level patterns that we find in the end. So this, I'm at the end of my talk. Thank you.
Okay, thank you, Daniel. Let's give Daniel and Sven a virtual hand for 2 very nice presentations. I would like to ask our attendees to type their questions in the question and answer box. Right now, I don't see any questions for either one, but which basically means I get the floor to ask Sven some questions for his presentation. Sven, this is going to your the beginning of your talk about the challenges for synthetic biology. And I want to try to tie this in with the the title of this eSymposium. It's been almost 20 years since the beginning of the field, right? So of synthetic biology, since the very early circuits. We still can't build very complicated circuits, right? With a large number of genes. If you look at a modern day, let's say microprocessor, and you look at complexity, the number of parts as a measure of complexity, it isn't really any simpler than a typical cell, yet we understand these things fully. On the other hand, for biology, I think the gap is still very vast. Part of the reason for that is perhaps the fact that, well, in engineering, we like to build things in a modular way, and this modularity helps us understand things better and build more complex things fairly quickly. Now, this brings me to the question, and also which touches on your goal or dream to make biotechnology from a discovery science to a true engineering discipline. Is biology modular? And if so, is it just a matter of time before we can design and build a working complex synthetic circuit? I mean, perhaps a hint comes from evolution. I mean, the fact that biology is evolvable could also imply that it's also modular, one could argue. So that would be good news for synthetic biologists. But I wanted to get your opinion on this. What are really the main obstacles for building very complex synthetic circuits, in your view?
I don't think there's much to answer. I think you already had it all in your question. So biology isn't modular. We need to accept that until people like you come up with design principles that allow us to deal intrinsically with noise and factors like that. And there seem to be some ideas around for certain aspects of it, but most probably not yet in a very, very broad, diverse sense. So there might be specific questions that we can deal with in a conceptually modular way. But I think fundamentally, this is our problem, and I don't see any easy way out of it. So I think synthetic biology was a bit over-optimistic at the beginning, which was very good because it got tons and tons of things started. But whether we will ever get to the dream of the motherboard with I don't know how many circuits, I think this is still up in the air.
In some sense, I'm surprised you actually your answer is biology is not modular. But if it's indeed not modular, then are we doing the wrong thing comparing it to engineering? I mean, a lot of the concepts that we use in building synthetic circuits draw directly from VLSI circuit design, electrical engineering, or mechanical systems, and so on. So if we can't rely on this modularity, perhaps we need a different direction, a different way of developing the field of synthetic biology. Maybe this is a question for later, but I was actually surprised by your answer.
I agree. Let's take it up after Wendell's talk.
Okay, one question then for Daniel. Somebody said, I'm not super familiar with the machine learning. Did the fact that some data point which are not well predicted by the software mean this difference is linked to some variable not linked to the DNA sequence?
So, I mean, the model is definitely not perfect. That's, and probably no model is perfect, but some are more perfect than others. And this might also mean that I missed in the construction of the model some features that either might not be DNA sequence linked, or that I did not extract from the DNA. This can both be, but I think I rather missed some features than that there might be some external features. I could not come up with now an idea what could be variables that are not linked to the DNA. Of course, especially since all the system components or the setup was the same for all experiments and for all variants. So the only thing that was changed was the DNA sequence, how the DNA sequence was arranged. But there might be also some mistakes, let's say, in the experimental setup that might influence this.
Okay, another very quick question to Daniel, and then we'll move on to the next speaker. So Daniel, can you comment how one can know protein copy numbers via the use of fluorescent proteins at each position in the operand?
Yeah, so I'm actually not looking at protein copy numbers here. The expression level, it's a very abstract axis label. I'm looking at relative fluorescence compared to a standard that I built before. That is a monocystronic standard. So what I'm actually looking at is how does a monocystronic construct compares to an operon construct. And in that way, I have kind of an internal or an experimental reference. What I looked at was really that all these standards for each of the fluorescent proteins have similar expression levels so that I can kind of compare it and there are similar properties, but I don't look really at the individual protein levels. This could be potentially done if you really purify it and do some experiments to do some standard curves. You could compare this, but this was not my main goal to look at the exact protein copy number here.
Okay, thank you very much. And thanks again to Sven and Daniel for very nice talks. This brings us to our feature presentation and our distinguished keynote speaker, Wendell. Let me introduce him, although he needs no introduction. So Professor Lim is Professor of Cellular and Molecular Pharmacology at the University of California in San Francisco. He's the director of the UCSF, UC Berkeley, NIH Nanomedicine Development Center and the director of SynBerg, the Synthetic Biology Research Center there. He earned his bachelor's in chemistry from Harvard and his PhD in biochemistry and biophysics from MIT under the guidance of Bob Sawyer. He then did his postdoc work with Frederick Richards at Yale. And during this postdoc work, he began to think about signal transduction and methods to evolve the module components of the proteins. His lab now focuses on constructing cell signaling systems from modular components to both study and engineer these systems. And he also serves as a mentor for the IGEM, International Genetically Engineered Machines competition. He has received many awards and recognitions. Let me just mention a few. The Byers Distinguished Professor at UCSF. Since 2008, he's been Howard Hughes Medical Institute investigator. He's a 2001 Kirsch Foundation medical investigator. He was named the David and Lucille Packard Fellow in Science and Engineering. And he's the recipient of the Hans Neurath Award from the Protein Society. And he was listed among the 50 people who will change the world by Wired SmartList magazine in 2012. It's a great pleasure and honor to welcome our keynote speaker, and I'd like to give him special thanks for getting up this early in the morning in California to be with us here today. Wendell, the floor is yours.
Okay, thank you. Okay, hold on. Could you start my video? I wasn't able to start it.
You mean your presentation?
Well, I was hoping to show my face also, and somehow it won't let me do that. But I'll go ahead and
okay,
start.
let me see what could be going on. So
the host has stopped my video.
Okay, let me see what could have happened.
Oh, okay. No, host is asking me to start my video. Great.
Okay.
Okay, good. You can see me.
There you are. Yeah. Okay.
Okay.
Good. Yeah.
Well, let me go ahead and start sharing. And okay, can you all see my slides?
Yes, we can. Okay. And your mouse.
Okay, wonderful. Well, so it's really a great pleasure to be able to visit you in Switzerland virtually. And I want to thank Mustafa and Sven for inviting me here. It's really a pleasure. I have great interactions with the people at the ETH. What I'd like to do is to talk about things that we've been doing in the last couple of years, trying to address this problem of trying to program cellular machines like this migrating T cell that you just saw a movie of, trying to engineer cells that could treat disease, build tissues, but really at its heart, one of the major goals is to try to elucidate what are really the fundamental design principles and some of the issues that Mustafa brought up about modularity in biology. Over the last several years, there's been this amazing explosion in immunotherapy and this idea that we can have, for example, CAR T cells. The first cell therapy was approved in 2017, but these are essentially cells that you can reprogram immune cells and put back into the body. And so this creates a really amazing challenge for us, which is we now have a platform in which we can ask, how do we actually engineer what these cells do, and what's the best way to design a cell that we can release safely into the body and then program them to carry out autonomous therapeutic action. So instead of, for example, like engineering a bacteria to be released in fields in the farm, this is the case of trying to release a cell inside our own bodies and program what it's going to do genetically, and do so, in a safe and controllable way. So we've been really excited about that, and so as I said, one of the big developments is this idea that you can redirect a T cell with a synthetic TCR, or a chimeric antigen receptor, to recognize a molecule that's presented on a cancer cell, and then have a T cell now kill it. You can take a patient's own cells, put this kind of synthetic receptor in, and then put it back into the patient. And in the case of certain blood cancers, you can get this remarkable effect of clearing the cancer. But although we view this as trying to make the best T cell, in a way, more recently, especially as we've tried to address problems in recognizing solid cancers, we realize that it's not so simple. Not only must the T cell be directed to kill the cancer, but it must also be very carefully engineered to avoid interaction with normal epithelial tissues, many of which share the same antigens on the surface as the cancer cells. Moreover, we know that in many solid cancers, there are cells that suppress the immune system. And so a T cell must be able to overcome or actively block that suppression. And then in addition, T cells and immune cells in general have sophisticated autocrine and paracrine communication systems by which they, for example, start to amplify themselves and lead to proliferation and expansion. They can increase their own dosage. And then in addition, really to be effective in many cases, T cells or immune cells need to be able to communicate with other cells, anti-tumor cells in the immune system and throughout the body, and call them in and have them be part of this ecosystem. So really, what I've come to realize is that immune engineering of these therapies is really not just a sort of a single cell effort, but really it's a complex multi-cell systems problem, trying to understand how your design cell actually interfaces with this whole existing ecosystem. One of the things I really want to emphasize in this talk is the idea that, and this is really kind of a hunch on our part, that we are sort of becoming very interested in not just engineering individual cells, but how we actually start thinking about controlling and engineering multicellular systems. And I tried to explain how even engineering a CAR T cell really is a multicellular problem. But of course, we know that in complex biological behaviors like development or regeneration, that genetic information is encoded in a cell or lineages of cells, but these are able to interact through at the multicellular level to create macroscopic structures or regenerate them. And that really at its heart, this involves many different scales of cell-cell communication involving morphogens, inhibitors, juxtacrine signaling, and adhesion. In addition, of course, many of the most interesting circuits in biology are ones that involve multiple cells, like our neurons or cells in our endocrine system. And what's shown here is just an example of, for example, what goes on in a pancreatic islet to regulate blood glucose. We of course think of the beta cell as a producer of insulin, but of course we know that in the islets there are these alpha cells and delta cells that antagonistically act on the beta cell, and it's really this network that is responsible for sensing and achieving the high level of homeostasis that we see in this case. So I want to try to address in this talk some of the things that Mustafa talked about. That one, we think that it may be a lot easier actually, and biology may use multicellular sort of circuits to achieve the kinds of circuits and adapting circuits that he spoke about. And that in terms of modularity, we are really starting to think that of course, where there is an obvious, in many ways, level of modularity is that biology is built of individual cells. And I guess what we would posit is, for many years, I've been obsessed with engineering a single cell as perfectly as possible, but it's very, very hard because you have all these molecules floating around, and it's hard to have enough orthogonal channels to really avoid one another and avoid, unexpected behaviors. I guess I feel like that the answer to how to get increasing complexity is to go to this higher level of multiple cells, where you can start reusing parts and you inside the cell, but then you have fewer sort of channels that you have to worry about in terms of how the cells talk to one another, especially when you start invoking spatial addressing and relationships between the cells that allow, and you have limits on the distance of communication. So just to summarize kind of this bigger point is that we think a lot of the interesting biology you see, certainly in metazoans and mammalian organisms, but also even in, say, pathogens or other things like that, involves really this hierarchy of molecular, single cell, and then multicellular systems. And that one of the things that we've been interested in is asking, what do we need to do at the synthetic biology level to facilely engineer the relationships between what cells say to one another? I personally have been, and many have been focused on how a single cell makes decisions, but we realize that certainly in metazoans, most of those decisions are what they're going to say to other cells. And so what are ways that we can start bridging this gap so we can look at multicellular or cell-cell interactions? A number of years ago, two really great postdocs, Leo Morsoot and Nicole Roybal, led our efforts to start trying to design orthogonal cell-cell signaling platforms. In this case, one of the most exciting ones was the synthetic Notch receptor, or SynNotch, which allows us to rewire cell-cell juxtacrine or neighboring communication. So the Notch receptor is involved in many different sort of developmental processes, and the Notch recognizes the delta ligand on neighboring cells. That engagement leads to a mechanical change that leads to cleavage of the intracellular part, which can then go into the nucleus and regulate transcription. So what we realized is that we could cut off the outside and the inside of Notch and put on artificial recognition domains like single chain antibodies that recognize whatever target molecule we want. We could also put on the inside synthetic transcription factors and then promoters that drive whatever genetic payload we want off of that. And in that way, we have the ability to reprogram a cell to sense novel environmental or neighboring inputs and then control whatever genetically encoded output we wanted. We found that we can put many different heads onto these receptors to recognize many different ligands. And at the same time, we can also put different transcriptional domains on the inside and different payloads. So we can make many different flavors of this. And these are intrinsically orthogonal. They don't crosstalk with either the native Notch or with one another when you put them in a cell. You are still limited by how much you can deliver to a cell, or at least intrinsically the molecules do not have crosstalk. Okay, so how do we start deploying these sorts of generic receptors into intelligent biological systems? And so one of the challenges that we first applied this to was the idea of how we can make a smarter CAR T cell, something that could recognize tumor versus normal much more precisely. So as I said, one of the big problems is that when we think about attacking solid cancers, that many of the antigens that are present on a solid cancer, they may be overexpressed, et cetera, but they exist on other normal epithelial cells at some low but detectable level. And so if you design a CAR T that would recognize either, in this case, the green circles or the purple triangles that are on the cancer cell, they might cross-react with these different normal bystander tissues. But let's say in this case that the cell has this combination of A and B is unique to the cancer and not present in any normal cells, then if we could make a cell that would perform Boolean logic that would, for example, require A and B, then we could get something that would be much more specific. So we decided to see whether we could do this. And one easy way to do this, or relatively simple way, was to actually try to design a T cell that actually had to go through multiple states, multiple state changes in order to become active and competent for killing. So the idea here is that we're going to take a T cell and we're going to put in the gene for a CAR, a killing receptor, that recognizes antigen B. But because this is going to be expressed not from a constitutive promoter, then that cell is what we call unarmed. It's not going to be able to kill an antigen with a cell with antigen B. But that promoter is going to be responsive to a SYNACH receptor that recognizes a different antigen, antigen A. So we're going to go through a sequence of multiple states. If the cell encounters in the unarmed state a target cell with antigen B, what it would normally kill, if it was a simple CAR T cell, then nothing will happen because it is unarmed. Okay, it doesn't express the CAR. If it were to encounter a cell with antigen A, now this would activate the SYNACH and lead to expression of the CAR. So now the cell would be primed or armed, but it still wouldn't get activated because there's no antigen B that the CAR recognizes. But if it encountered a cell with antigen A and antigen B, then it would get primed and then recognize the antigen B through the expressed CAR and then be able to get activated. So I'm going to skip over this. So basically, we showed that this could work, in fact, in vitro. But just to remind you in this animation, what we're trying to do is say, if we put it into a body, a patient, or an organism, the problem we're dealing with is that a tumor might overexpress this red antigen, and you could have a CAR T that would kill it, but it would also kill normal cells that express low levels of that antigen. The idea here is that in this new design, we have a T cell that will, in the tumor, recognize a priming antigen, this green antigen, and then in response, only transiently and locally induce the expression of the killing receptor, the CAR, and now be able to kill those cancer cells. If that cell, those T cells now go into the normal tissue, where in this case there is no priming antigen, then it's inert, and even though the killing antigen is there, it can't kill. So it really has to have this sort of dual authentication kind of sequence of events to happen and get activated. Okay, so what we did is we made these cells, and I said tested them in vitro, and then wanted to test them in mice. So what we typically do is we make a dual tumor model where we have the tumor has two antigens, whereas the bystander only has the killing antigen. And then we put the T cells in the tail, let them go everywhere. The hope was that when they entered into the bystander model, that because there's no priming antigen, the T cells would be inert. But if they entered into the tumor where there is the priming antigen, they would become locally primed and be able to kill those tumor cells and shrink it. But of course, we were worried about multiple problems. One, would this transient induction of the CAR receptor be sufficient to lead to a strong response and to be able to clear the tumor? But then on the other side, there's the issue of if the cells were primed, could they actually then move back to the other bystander tissue and exhibit toxicity? So we did this experiment, and we've done this now in multiple tumors. But what we found, what Cole found, was that, in fact, in every animal, what we saw is that the dual antigen tumor was cleared quite rapidly, but then the antigen tumor in the same animal exhibited growth as if there were no T cells there. And that's shown, for example, down at the bottom here in this example of this mouse, where the T cells that we put in are able to completely clear the dual antigen tumor on the right side, but then completely ignore in the same animal the single antigen tumor, even though it's the same antigen that you kill with is present. So we see this precise localized activation of T cells that's really quite remarkable. More recently, and this work led by Jasper Williams, a former student, we have talked about how these SYNACHs are very modular. Well, we've asked, can we start combining multiple SYNACHs and CARs and TCRs to make different kinds of logic gates and recognition circuits? And so beyond the single antigen recognition, as well as the AND gate that I showed here. And I won't go over all of these, but basically what Jasper and colleagues have shown is that we can start linking these systems together to make a huge variety of different possible gates and really have a whole almost periodic table or catalog of recognition circuits that we have available to recognize from 1 to 4 different antigens, including both positive and negative logic. We can have negative not antigens, as well as things that incorporate and, or, et cetera, and again up to really 3 or 4 antigens. Just want to go through a few examples. How can we integrate something that would have a not recognition? So this would be useful in the case where, for example, the tumor in a normal tissue might share a antigen that could be used for killing, but there's also an additional antigen that's only present in the normal. So there's something that you'd like to actually turn off the T cell. And so what Jasper made here is he took an AND gate that recognizes the surface antigens GFP and CD19 through the AND gate that I described before, but then in this case also put in another SYN notch that induced proteins that are involved in apoptosis that hopefully could be an override. So they did shown here is a different scheme where you have one SYN notch turning on the CAR and then another SYN notch, and these have different DNA binding domains that turns on TVID, which is a pro-apoptotic protein that ideally would prevent one of the key responses, which is proliferation and expansion of the T cells. So you have this antagonistic function. And what he shows is that looking at target cell killing in vitro is that if we have GFP and CD19, we get efficient killing, but then if you add HER2, that will abrogate the killing. And then we say the same thing for T cell proliferation. So these are plots where a dye is diluted with each division. So the many peaks indicates a lot of division. We see that with the dual antigen target cells, but if you now add HER2, that overrides that and shuts that down. So we can override killing and proliferation in this way. And then here's another example of really trying to daisy chain these circuits together to get a 3-input AND gate for really ultra precise targeting. So here we use these antigens EGFR, HER2, and MET. And so this circuit here, and there are different ways that we can do this, but it's really an in-series circuit where we have, just shown at the bottom here, we're going to have antigen 1 activate SYN notch 1, and then that's going to lead to expression of SYN notch 2 that could now be triggered by antigen 2, and that would lead to expression of the CAR that could recognize and kill based on antigen 3. So it's a 3-receptor cascade. And so what we find here is that we see almost no killing with any of the targets that have 1 or 2 antigens, but only efficient killing when we have target cells that have all 3 of that. Same thing for proliferation. We see high all or non-proliferation with the triple antigen target cells, but none with the singles or doubles, even though these all contain HER2, which is the antigen that's being targeted by the CAR. And then we can put these similar tumors in bilateral mouse models where we have both of them have HER2, but one has the 3 antigens, one has just the 2. And what we see here is that these cells will ignore the 2 antigen tumors, but efficiently kill the 3 antigen tumors. So we can get this really remarkable level of specificity. I should point out that, as you might expect, this takes longer, several hours longer to activate, but I think you get, the trade-off is that longer decision-making by the cells leads to a tighter, more specific decision. Okay. Another example I just want to show you is work done by Rogelio Hernandez-Lopez, another postdoc, where he's really been focused on trying to engineer cells that can sense antigen density, not just combinations. And so, for example, in the case of the antigen HER2, it's a tumor-associated antigen that's overexpressed in many classes of tumors, but HER2 is expressed at low levels in many normal tissues. So HER2 CARs have shown a lot of toxicity. And so what we really would like to do is to be able to have much more control over this. We'd like to be able to have CAR T cells that can ignore normal levels, but then in the tumor. And so what Wendell came up with is a simple idea that why don't we combine a SynNotch and a CAR that both recognize the same antigen. But in this case, we're going to use a low affinity SynNotch, something that is sort of a high density pre-filter. It won't get triggered until there's high density of the HER2 antigen, and then have that now turn on a high affinity CAR, which would really amplify the output, with the idea that this would now give us more of an all or none behavior like this ultrasensitivity. And in fact, as shown here, if he takes these and looks, and he constructed a whole set of cells with different levels of HER2 antigen, that if we look at what happens with, say, a low affinity CAR, it gives us linear behavior, but we can get this tight thresholding with this low high cascade. So we can get a nonlinear response by combining these and kind of filtering it in this cascade in this particular way. This just shows that he's able to take, in this case, spheroids that have made up of the high or low HER2 cells. And then in this case, he's using a caste-based dye in green to show when there's killing. And so we start with these low and high HER2 spheroids. And then what you see is after 72 hours, after we add the T cells, that a low affinity HER2 CAR, just a straight CAR, will disrupt and kill all the disrupt the spheroids and lead to death. But the circuit that we talked about, that this ultrasensitive one, leaves the low HER2 alone, but still disrupts and kills the high HER2 spheroids. Moreover, we can go in vivo and implant low and high density HER2s, and we can see that there's, and these scores of 1+ and 3+ refer to kind of clinical scales of HER2 expression that pathologists use, and that we get really clean discrimination killing of the 3+ HER2 tumor and sparing of the 1+ HER2. Okay, so one other thing I want to point out is just how spatially precise this killing is. So one of the things that Rogelio did is to create this assay where he puts a spheroid of a high HER2 in Matrigel, and then surrounds it with an excess of low HER2 spheroids, shown in pink here, and those are what are circled here in red, and then puts in T cells and looks for killing. And so what he sees is that there's a lot of killing, of course, in the high HER2 spheroid, but what we see is almost no killing in the surrounding spheroids. And what we can plot this and see that really by 0.1 millimeters, which is shown here, there's almost no killing of the low HER2 spheroids. So even though the T cells are free to migrate between these spheroids, that killing drops off significantly within this distance of 0.1 in this particular millimeters within this assay. So we think it's really pretty amazing that you can get this highly concentrated expansion and killing, but then really just within less than a millimeter, you're not getting bystander killing. We think that obviously the sigmoidal recognition curve that we put in is part of this, but we also think there may be other things such as the induction of adhesion molecules, as well as the induction of local cytokines that really help control this highly focused type of killing that immune cells are capable of. Okay, so just to summarize, in terms of deploying these circuits in therapeutic T cells, we've been using systems of juxtacrine receptors and shown that they can be used to achieve high selectivity of tumors with specific antigen combinations, many different kinds of recognition circuits, as well as different densities. I didn't talk about this, but we are developing now multi-antigen circuit designs for specific cancers, including glioblastoma, pancreatic cancer, and a number of other ones. And we've done a lot of bioinformatic analysis that I refer to in this paper to try to identify what are specific tumor combinations that are most informative and that we can target. And these are actually going quite well, and we hope to get into clinical trials quite soon. We're also designing T cells that can inducibly deliver cytokines and chemokine payloads that can help to remodel the tumor microenvironment in other ways. But basically, we're really excited about how these tools that involve cell-cell communication can really help us in attacking solid cancers. really demonstrates, we think, the power of having a cellular agent that is able to integrate into and interrogate the tumor ecosystem, combined with the ability to be able to program its recognition. Okay, so I want to talk about a different area that we've been interested in too, which is this idea of synthetic development. Can we understand the basic rules of development? What its primitive operations are? Can we start not only trying to understand those rules, but really try to engineer how those sort of components and the circuits can lead to new self-organizing multicellular morphologies. And so this is work that was pioneered by both Leo Morsood and Satoshi Toda. Both of them have their own labs now. What they realized is that there are many different circuits for controlling self-organization in development, but one of the unifying themes is that cells recognize one another through cell-cell interactions, and then those interactions lead to morphological changes. They could change what cells adhere to one another, how cells move, what states they differentiate into, or even produce more signals so that you could have cascades or feedback that would come back. And I should point out that spatial reorganization can also change what cells next to one another so that you could get new kinds of relationships evolving in a series of events. And so what this trying to link this sort of cell-cell communication with morphological changes, we realized that we could use SYNAPse receptors to create new kinds of relationships between cells, and then use those to control the expression of, for example, cadherins that are involved in cell sorting and adhesion, different fluorescent proteins as markers of different states, as well as new SYNAPse ligands that could feedback and give you cascades of events. And so what they did is to put these into these L929 fibroblasts that don't self-organize, and then ask which ones could actually self-organize. I refer you to this paper from 2018, but just to show you a few examples, these are some movies of cells that these are two species of cells we put together, and then what happens is they actually can start forming, in this case, a 3-layer structure or a polar structure with a green and a red pole. And then this is one where they should form concentric layers of green in the center, and then red in the middle, and then blue on the outside. These are all movies that take about 24 hours to take place. And then this is one that has these multiple sort of arms, green arms that are generated. So just to kind of go over an example here, this is a case of a 3-layer circuit where we made 2 cells, A and B, where A will stimulate B through a SYNAPse and induce the expression of both GFP and high levels of E-cadherin. So those cadherins will lead to green cells forming and then collecting in the middle. It's an example of phase separation at the cellular level. And then in this case, we also have reciprocal signaling. The GFP ligand that's produced is an extracellular version that is recognized by SYNAPse in the first cell, which leads to expression of M-cherry and low levels of E-cadherin. So these now turn red and then sort to the middle edge, and they're right there at the interface between the green and the blue cells. So I won't go over this, but we were able to use these components and build many different kinds of circuits that lead to different kinds of morphologies with this fairly limited toolkit of components. And they're basically, these systems are able to self-organize in a stereotyped sequence of events, just like normal developmental pathways. They can form discrete compartments and layers, and they can also drive the emergence of increased numbers of cell types. We have more phenotypes than the starting number of cell genotypes, and then we can actually cut these up and they can actually self-repair when damaged, and we can get both symmetric or asymmetric structures forming. Okay, so what I've shown you is work where we look at juxtaposed signaling cells that are right next to each other. But we know that another really important part of spatial organization is morphogens, or things that actually provide positional information. That oftentimes we will have a cell that really is a landmark, and then other cells know how far they are from that cell. And that's usually done through these morphogens, things like hedgehog, Wnt, BMP, etc., that diffuse from those landmark poles, and then cells can detect how far they are because they form this kind of gradient. So we're very interested in asking whether we could not only make short range, direct contact mediated signaling like with SYNACH, but could we make diffusible or longer range signals that could allow us to start exploring and playing with these sorts of morphogen-like gradients that can measure distance and give relative positional information. Okay, so one of the things I want to just emphasize is that in the normal SYNACH system that we can activate with, say, a membrane-tethered ligand like GFP, but if you actually have soluble GFP, it doesn't actually activate, and we think that's because of the need for this kind of mechanical force on the receptor. But what Satoshi showed is that if he actually made, for example, soluble GFP, but then constructed not only a SYNACH that can recognize it, but also an anchor cell with an anchor molecule that recognized a different interface, then we could actually create kind of this sandwich where the anchor cell would present a tethered molecule of GFP to the receiver cell. And what he found is that, in fact, when he has both the anchor and the receiver and the secretor cells, only then does he actually get activation of, in this case, M-cherry output. So we can actually make a system that will trap a diffusible molecule and then lead to activation of this kind of synthetic receptor. And I should point out this, in related work, JP Vincent's lab at the Crick Institute has also shown that they can turn GFP into a synthetic morphogen, and actually works in Drosophila, work that we just published back-to-back recently. But so to look at this, our system, what Satoshi did is he reconstituted an in vitro kind of pole and body. These are two patches of cells. There's going to be a patch of pole cells that will secrete GFP in this case, and then a patch of body cells that have both composed of multiple cells, in this case, both anchor and receiver cells, and we can mix them together in different ways and change them. And then he puts a agarose layer over these so that we don't get turbulent flow and diffusion. And so if we do this with a membrane-tethered GFP, what we see is that activation only occurs right in the edge of the body that is next to the pole. But if we do this now with a mixing together anchor and receiver cells in the body, and then having a secretor cell, that we can get this gradient of response. This is the M-cherry response from the receiver cell that gives us now positional information. One of the things he's shown is there are multiple ways to reshape this morphogen gradient. I won't go over this in too much detail, but basically he's able to change, for example, the density of the anchor proteins and cells, and this can shape what the gradient looks like, whether it's if there are many anchor molecules close, then it will trap most of them close by, get a sharper gradient, but if they're a lower density, it can spread out further. In addition, he's shown that we can have, for example, use inhibitors. We can make a version of an antibody like the receptors, but one that's soluble that would counteract and block either anchor binding or receptor binding. And in this case, for example, he can make a second pole on the opposite side that makes one of these inhibitors, and this can lead to dramatic reshaping of the resulting and sharpening of the resulting gradient response that we see. And this is very typical. Almost all morphogens have inhibitors that counteract them in this sort of way. Okay, so one of the exciting things is that not only can we make these connections, but because of the modularity of SYNACH and the way that we can reconnect the output to different things, we can start building more complex circuits. So one example is to build a positive feedback circuit where we have the body recognizes GFP, but then in response, what it does is it makes not only M-cherry, but it also makes more GFP. So you can have propagation. And so what's shown down here is a movie of what happens with no feedback and the response that you see with the positive feedback on the right, that you see we get amplification and propagation across and get this kind of square wave function across the field of the body by introducing the positive feedback. We can also introduce negative feedback. So this is a case where we have the SYNACH that receives the GFP now makes its own inhibitor. And what this leads to is shown, let me go to the bottom right here, which is that if you look at the shape of the gradient, first of all, it forms much faster, but then also what we get is that the gradient is much more stable with time and with sort of amount of morphogen that's produced. And this is something that interestingly Nama Barkha has shown in native circuits, developmental circuits, that if you have negative feedback, it can lead to a much more robust and stable morphogen gradient. In those cases, it's things like degradation of the morphogen, even though this has a very different mechanism, in the end, it leads to the same sort of overall behavior of a more stable gradient. Okay, so I've shown you positive and negative, but basically, we have multiple morphogens now. We can make one out of M-cherry, we can make one out of GFP, other molecules, and then inhibitors, but then we can link them together. I've shown you about positive and negative feedback, but we've started to link them together in different ways, morphogen cascades, etc. And this now allows us to do different things. I showed you before taking a normal gradient, but then putting positive feedback to shape that into a more square wave. If we now add an inhibitor on the other side, we can get something that's more of this truncated stripe. And then we can build things like this shown here. One of the classic examples of a developmental model are a multi-striped, or the so-called French flag model that Louis Wilpert described as a kind of classic model of development that has 3 domains. And we can build, there are many ways that you could do this, but one of the ways that we've been able to do that successfully is we have a morphogen that produces, pole that produces an M-cherry morphogen. And this is activated, and one of the things that it does is that it leads to expression of a second morphogen, GFP. So that's a 2 morphogen cascade. The GFP itself is also induces more GFP, so there's a positive feedback loop. And then we have an inhibitor pole that produces something that blocks the M-cherry. So we're combining many different elements here in the same sort of body and system. And so what we show here is that we can track this, and what we see is that BFE is a reporter for the M-cherry response. We get this first stripe here, but then because of that induces GFP, and there's this positive feedback propagation, we get a second stripe with GFP, but then both of these are inhibited by the inhibitor shown on this side. So we can get this kind of 3-stripe behavior, and then many other kinds of patterns that we can get. So just to summarize this part of the talk, we've shown that we can turn multiple arbitrary proteins into diffusible morphogens, GFP, M-cherry, but many other things, including molecules that occur naturally. First of all, we think that's interesting and relevant to sort of this puzzle of how these really important molecules like morphogens could have arisen and evolved. And we actually think it's not that hard, as long as you have some way of trapping these morphogens, whether it's on a cell surface or on the extracellular matrix, and then receptors that can detect it. With these systems, especially using a SYNASH version of this, we can program diverse combinatorial morphogen interpretation circuits, because we can start linking them together in different ways. As I said, this gives us some insight on how current morphogens may have evolved, but we're also really interested in the idea that can we start program cells to recognize endogenous signals, ones involved, for example, in injury and inflammation, and convert them into morphogens so that they can provide positional information to program, for example, regeneration. Okay, so as I said before, we have become really interested in, if you asked any biologists, of course, they would say it's obvious that cells themselves and multiple cells present a form of modularity. And I think that that's true, it's pretty obvious, but I think that it's really, we're really excited about trying to provide tools or develop tools that allow us to really play more with the relationship between multiple cells, and leads to the question of what is it that's really necessary to build multicellular like systems. And some of the tools that we have been trying to work on are, of course, things that are involved in immediate juxtacrine communication, like SYNASH, things that can have action at a distance and provide positional information like the synthetic morphogens, but also we're working hard on synthetic adhesion molecules that can bind to either, where we can program what cells bind to one another, or to things like ECM, but that can tunably sort into different sorts of structures as well as provide different kinds of mechanical properties. And we do think that when you combine spatial relationships that are determined by things like adhesion with spatially controlled signaling, you really are going to be able to get a lot more sophistication and control. So in all these cases, we're trying to develop modular parts that can construct new cell-cell connections without crosstalk, and then deploy them to create synthetic systems. We're moving forward and trying to construct more complex synthetic tissues. One example is trying to guide development, for example, with organoids, rather than just stimulating them with cocktails and a protocol of different soluble factors. We're actually trying to build synthetic organizers or niches, cells that bind to and organize around the progenitor cells, and actually provide them with spatially encoded information that could more precisely lead to their development. As I said before, we're also interested in this idea that we could program our regeneration in situ. Can we recognize signals of injury and actually use those as landmarks? Can we make adapter cells that could recognize those and convert those into, for example, things that would control regeneration? And very similar to that is the idea that could we engineer more sophisticated immune cells that could physically recognize and then reorganize tissues, either in the case of damage or in the case of inflammation, but also in the case of tumors, where they really want to infiltrate and really remodel what's going on and the relationships with different cells. And so with that, let me end by thanking the amazing people in my lab who have done all of this work, including Leo, Cole, Satoshi, Jasper, and Rogelio, and many others. I want to just say that we're really excited about both synthetic biology coming to fruition with some really exciting medical applications, as in these engineered T cells, but really also trying to understand these basic rules of the different hierarchies and modularity of biology. We have a new institute, the Cell Design Institute, which is really focused on these issues, both very fundamental design principles as well as different applications. And if anyone's excited about joining us, please contact us. We're always excited to have great new people. So thank you very much, and happy to answer questions.
Thank you very much, Wendell. Very nice talk. I would like to ask the listeners to start typing in their questions. There are already a couple of questions. Let me start with one question. That was kind of very intriguing, how you could shape these morphogen gradients. So what you've shown that with some combination of positive feedback, negative feedback, also inhibitors, you can shape these gradients to finally stationary patterns. Have you thought about designs that will give you more dynamic patterns? So, for example, like traveling waves or different types of spatial patterns that can come from these capabilities of designing these notches?
Yeah, so we have, and it's known that with native notch you can get traveling waves, and of course one of the most famous things that happens in development are these oscillators that can lead to somite formation, and so that's it's clear. I think we're trying to get a better I mean, we're limited by things like the lifetime of the whatever effector we produce and how long that's going to stay around. So and then as you know, when you start trying to make them degrade faster, you lose signal and all that. So I think we've so the question is, how do we I mean, I think the answer is that we're trying to have effectors that we can actually tune, both change the lifetime of the induced state change, either to make it shorter or longer, so that we can start looking at these sorts of things. And I would say that the other side is also equally interesting, that these are all, most of the systems that we've looked at are relatively sort of steady state, whereas in real development, of course, many things after, sort of, a lot of consideration, certain cells will undergo irreversible changes. And that's a critical part for divergence of cell fate.
Okay. I have a question from one of the audience members. Since biological reactions are stochastic, combining several logic gates should amplify the variability at each step. Is there a limit to the number of processing steps that can be combined in the CAR T cells? And have you looked at the loss of signal along these circuits?
Yeah, I mean, so that's a great question. In this paper that is coming out this week in Science, we've looked at some of that. It's difficult for us to get much more than 3 circuits in there, but we have built both cascades in series and parallel triple AND gates, and also we have one stronger promoter and one weaker one, and it definitely the signal, there is some decrease in signal. And if we have to put the stronger one first, et cetera. So you're right, there is some limit, but I mean, I think an important thing to remember with the T cells is they have this built-in sort of positive feedback amplifier at the end. So once you really kind of get to that stage and you recognize something and they can start producing IL-2 and cytokines like that, they kind of can ramp up and proliferate and do that. So I think a lot of that's really one of the magical things here, is putting a lot of, all these are, have in common a lot of filters, but then once you get to the right decision, really turning on the positive feedback.
Okay, next question is, as you have 2 different morphogens, GFP and M-cherry, and what you could tune the diffusion properties of them, did you try to reproduce some Turing pattern systems? I guess that's related to an earlier question.
Yeah. So we have to try to get things where you have local local activation, global inhibition, things like that, and so we have we are doing that, and the work is I think we have some promising results. It is tied, as you said, to the how quickly reversible those are. And so we definitely we're getting spots, is what I would say right now, but that's something that we're playing with.
Okay, very good. Back to the cancer, the CAR T cells, what are some of the regulatory hurdles that you would expect, for actually putting these things into the clinic, and you're having real patients using these type of synthetic cells?
Right. So that's a great question. So in this case, regulatory meaning government agencies and the FDA. And so as well as there's sort of commercial and economic hurdles. So first of all, in terms of today, you really, there's a limited payload of what you can put in with one lentivirus, which is the traditional way of doing it. But we do, when we're thinking practically about how to get something into the clinic, we are definitely not on the edge of pushing 3-4 antigens and things like that. We are trying to find sort of 2 receptor circuits and maybe a payload that we can fit in, that where we really think, and we do believe, that just adding another 1 or 2 elements will allow us to do much better, particularly in solid cancers. So we're definitely thinking very carefully about that. Everything is changing so much. There's a lot of work on so-called allogeneic cells, where you can sort of suppress the mechanisms of immune reaction of the receiver of the cells, so that you could use a universal donor. This would allow us to actually put more things and play around with them more. So there's some flexibility there in the future, but we're trying to have impact right now. So we're very concerned with keeping these as simple as possible, just adding elements that we think are going to give us the most bang for their buck. That being said, I want to just talk about the other side of that too, which is in the end, we think about sort of multicellular systems, and also how much what's the load on any cell. And I want to point out that the immune system really distributes its functionality in a lot of different ways, and we're very interested in developing multicellular systems where maybe you have sensing cells and then responder cells, and it's really a mixture of those that you put in. That's very antithetical to current regulatory desires, though I think one of the things that you could have is to be in that situation where you have vetted and tested the individual cells, but then now you put them together, and that so they, in some future, I think that could be more open up new doors.
Okay, thank you.
Wendell, maybe I can throw the question at you that Mustafa threw at me after my talk. How modular is biology? If I remember your scientific palmaris over the last 20 years, right? I think there were lots and lots of systems that actually were quite modular that you exploited to excellent benefit. Also see recent examples as presented. So maybe you have a different opinion than the critical biology is not really modular that I a bit respectless answer to Mustafa.
Well, so you know, I'd say that I mean, it's modular to an extent, and it's modular at different hierarchical scales. You know, and you know, I mean, evolution has a constant push between maintaining modularity and achieving optimality, right? So there's a push and pull of during evolution of what remnants of modularity you see. You know, I mean, there's definitely a lot of modularity we know at the molecular level. We can mix and match components and put, you know, genes behind different promoters. So there clearly is modularity, but you know, I've spent most of my life engineering molecules, and you know, it's very, very hard, I have to say. And what I do think is that I found it to be much easier and much more sort of truly modular to change what cells, you know, say to one another, or even, for example, in some of those experiments where we put different cells in the body, we're just putting different mixtures of the same cells in different cases, and are able to generate systems that do different and interesting things in a much more predictable way. And I, you know, the point is just the numbers, right? How many molecules are in a cell versus how many molecules are on the surface of a cell, and that can mediate real, you know, communication. And it's, I mean, I think that's part of why the cell is, you know, that we're not, life isn't just a bag of molecules, but has these components. And then again, the fact that, you know, for example, I mean, Notch and Wnt and all, they're used all over the place, right? But we don't have to reinvent them because of spatial addressing. And if you talk about getting to real circuits and like, you know, computer chips and whatnot, that's a key thing. I mean, what if we could make computer chips that self-organize their architecture, right? That's the brilliant thing. So.
Okay, I could take another question from one of the listeners. Thank you for the nice presentation. I have several questions about CAR T cells. Would tumor cells escape from T cells more easily if T cells are engineered with complicated logic gates? And would there be an easy way to tune the threshold of ultrasensitive CAR T cells? How would you approach a good balance between the off-target effects and tumor antigen escape?
Okay, so those are great questions, and I was waiting for that question to come, and but I'll briefly say that we are able to tune the ultrasensitivity by tuning, you know, both the affinities of both the SINASH and the CAR, although, you know, there are some limits to that, because as you tune them certain ways, your total sort of response amplitude will go down too. So you kind of have to find the right affinities to do that. So, but let me address this other question about antigen escape. So, right, yes, we know that if you design a drug or any drug that targets a certain molecule in cancer, that inevitably you get escape, and that's why most best therapies even don't last forever. Okay. And so then if you say, oh, let's take, now you require 3 antigens, right? Well, that's going to give you a lot more possibilities of escape, right? Okay, but it turns out that the systems that we've built actually are not susceptible to that. And they can actually deal with escape or antigen heterogeneity at the level of the SINASH, the priming antigens. Let's just take a SINASH to CAR and gate. That what we find is that not every cell in a tumor has to have the priming antigen. If there's a high enough number of them, percentage, then the SINASHs can get activated, the cells on the T cells, and then you can get what we call trans killing, that it can then kill a neighboring cell that has the killing antigen, okay? And so if you go down from, 90% to 80% of antigen A, the primary one, it really has no effect. Okay. And so really because the SynNotch is sort of this relationship in this network of cells that has a certain distance, but it is, the SynNotch circuits are able to integrate over multiple cells. So we think that's actually a really, we've shown that now. We've looked at heterogeneous tumors where we can kind of control how much of the primary antigen there is, and they are very robust to those differences. And so you can't get away from the escape by the killing antigen being deleted, and so you still want to try to make that something that's tied to tumorigenesis. That's required, and where you really can hammer with a really strong response, or maybe have two antigens that you kill, and we can do that with OR gates. But I think we think it's a really big advance that you can overcome the heterogeneity with some of those antigens.
Okay, we'll take a question from outside of ETH, from Tom Ellis, I guess at Imperial. On synthetic morphology, what genetic tools and functionalities do we need to develop and add to the ones you've already shown to be able to make more complexity?
Yeah, I mean, so, I've talked about how I think that's an open question, and I invite everyone to think about that. But I would say some of the things that we're thinking about are, as I said, synthetic adhesion and how we cause cells to recognize one another and stick to one another, and what kind of interfaces they form. And then, because if you think about a neuron, it has sort of an input interface and an output interface. These are the kind of things we'd like to be able to build. But also, I think, one of the big mysteries is all of this development stuff takes place where you use things like matrixel and some sort of matrix. And that's fairly poorly controlled. And really, the reality is we know that our own cells are making these matrices and remodeling them, having better control over that, because it's clearly, whether it's a cell wall or an extracellular matrix, you need to have this kind of mechanical structure to start building things and a framework for building your chips or whatever. So those are things. But then, of course, there's a lot more communication channels, whether it's channels or gap junctions or other things and then, as I said before, being able to control the dynamics, both making things faster, but also slower, so you can have permanent, memory changes along the way, either at the single cell level or at a circuit level. But I think it's an open question. What are the sort of primitives of these sorts of systems? And we can be inspired by what we see in nature, but there may be things that lie outside of that too.
Okay, thank you very much. Let me take one more question. Have you ever tried to express both SynNotch receptor and ligand in the same cell to produce something like a checkerboard pattern through cross inhibition, or if there's a reason why this cannot be done?
Yeah, so that's a great question. One of the problems is that we do see cis inhibition, where a cell will inhibit its own SynNotch through that way. What we tend to do is to do that kind of checkerboard and things like the Turing patterns is we'll tend to use a 2-cell system to achieve, for example, feedback. It means that we have to mix multiple cells together, but that's the way that we can do it. Or actually, it looks like some of these soluble forms work, where you could have the anchor and the receptor. Really depends on the geometries of these and what's going to favor a trans versus a cis interaction.
Okay, thank you very much. I think this is all. These are all the questions I've had from the listeners, and perhaps this is a good place to end this eSymposium. I would like to begin by thanking all the speakers for their nice talks, and thank you, Wendell, for this very intriguing presentation. I want to also thank Randy Platt, whose idea was to create these eSymposia. Also Alvin Hendricks and Carolyn Arndt for helping set this up, and my partner in crime, Sven Panke, for doing something on synthetic biology, and all the other speakers. I also wish to thank all the listeners, all of you who have stayed late and stayed up, also for you, Wendell, for getting up so early. I want to give you a very special thank you and a good morning to you from all of us here in Switzerland, and happy Thanksgiving. I guess this week is Thanksgiving in the US. So have a good day. Thanks everybody, and see you all later at the next eSymposium, hopefully.
Thank you very much.